my name is Amelie H. I am in grade 12 STEM, and today I will be talking about local learning and how that pertains to my capstone project of testing the waters of Still Creek to see how the pollution levels change in different areas of it. The hypothesis that my partner Mai L and I were working off of was that runoff from industry and other projects would introduce possible contaminants and that levels would increase as you head downstream. We measured the water quality along Still Creek and its potential effect on wildlife and habitat restoration. We tested for 16 different contaminants shown here on the screen. Ultimately, there was no consistent trend in any of the contaminants. The levels varied throughout the length of Still Creek. The only consistent change was in pH, which decreased steadily in a downstream direction towards Burnaby Lake. The conclusion that we reached from this data is that the contamination level changes aren't from natural causes, but from man-made ones, such as storm drains, construction, railways, businesses, garbage, and others. Of all the things to study, why this? We had wanted to do a more environmentally focused project. The city of Vancouver had started a project to uncover the creek in 2002 in an effort to bring back the wildlife and the salmon. We wanted to see if the water was as nice and clean as they had wanted. I'm going into environmental sciences, and we thought that this would be a good starting point for that. We also wanted something local that is easier to research and relate to than, say, how climate change is affecting trees. We thought that this would be a fun project, and it was something that we could do and continue outside the classroom. You might be wondering, why is this relevant? Well, Vancouver went through all the trouble of uncovering Still Creek in 2002 after 80 years to bring back the wildlife and the salmon. But there can't be wildlife if the water quality is bad. The chart that is shown on screen is lead. Lead is measured in milligrams per liter. Other than 3 instances, we measured the lead content to be at 20 milligrams per liter. When consuming water, there shouldn't be any lead in it. When we were collecting samples, we saw very few aquatic animals. There were, of course, the typical birds you see around Vancouver, but we only saw fish on one day and in just one spot. We saw mallard ducks, wood ducks, and Canadian geese. One memorable time, we even saw a blue heron. I really enjoyed this project because we were able to take what we learned in this project and apply it in our local environment. Local learning is like field trips, but on a much grander scale. We take what we have learned and apply it in our projects, either generally like coding or more local and focused, such as in this project. This way we get to experience the real world, see what works and what doesn't. For example, in this project, we had to learn on the fly how to collect samples on a rainy day and not have the rain affect our samples after we had taken them. A big reason for collecting our own data is that until going out and experiencing something ourselves, we have no frame of reference, no idea what the numbers mean and correlate to. There may also be hidden things that wouldn't be noticed otherwise, such as the fact that we did end up seeing fish on one day, ducks, geese, and a heron on the last day. We were also able to see the surrounding environment and pull conclusions for that. Why were the cyanuric acid levels so high at our 4th testing site? Maybe it was because there was a big pileup of garbage in the stream 10 steps down. Another reason is that we couldn't find any accessible data online. On the first day, though, we saw some students from Langara College doing the same project. They started in December. Through this project, we learned how to gather data from various sites in a variety of conditions. We learned how to make an interactive graph displaying all the data we collected in a way that makes sense, and were able to filter it without having to make 30 different charts. We had learned the theory of this in class at different times. However, this was the first time that we were really able to learn it, put it into practice, and see what actually worked. I thoroughly enjoyed the opportunity to get out of the classroom and get some hands-on experience. Continuing onward, we would like to eventually petition the city to clean and better take care of Still Creek. To do this, though, we would like to do more extensive testing, both in regards to more testing and more things to test for, such as E. coli and other bacteria. We would also like to try and figure out what causes the different levels of contamination, given that they change oddly. Thank you for watching. I hope you liked my presentation and enjoyed the other presentations as well.